Welcome back everyone. Now we'll be discussing about the second point of biological uptake process, that is the transport across the epithelial barrier. Now before going into this, I'll be just discussing you about the epithelial barrier. Now the epithelial, when we talk about oral administration, we basically think about the intestinal epithelial barrier. The gastrointestinal tract is designed to impede the entry of the pathogens, toxins as well as undigested macromolecules, while simultaneously digesting and selectively absorbing essential nutrients, vitamins and cofactors. Unlike the skin, which has keratin to provide an overwhelming physical barrier against the bacterial entry, the intestinal mucosa uses the biochemical and physiological mechanisms to complement its physical barrier to prevent the entry of unwanted materials. It is this protective mechanism of the GI tract that must be judiciously modified or circumvented for oral drug delivery. These obstacles include the hostile acidic environment of the stomach, the high enzymatic activity of small intestine, and the inherently low permeability of the intestinal mucosa, as well as the binding capacities of the resident mucus and luminal contents, the efflux pathways back into the gut lumen following uptake into the epithelial cells, and first pass metabolism by the liver. These physiological factors must be taken into account when designing in vitro tests such as dissolution test for solid oral dosage form, as well as other solid oral dosage forms like the various tablet, capsules, or even other solid oral dosage forms. Absorption of the solutes in the gastrointestinal tract occurs primarily in the three sections of the small intestine, that is duodenum, jejunum and ileum, and in the large intestine, basically in the colon. Each of these segments has unique anatomical, biochemical and physiological properties that dictate the parameters of digestion and solute absorption. The internal surface of the small intestine has three types of irregularities or projections, which increases the potential surface area for digestion and absorption. Macroscopic valve-like folds called the circular folds encircling the inside of the intestinal lumen are estimated to increase the surface area of small intestine threefold. Villi are tiny finger-like projections barely visible to the human eye, which increases the surface area by approximately 30 folds. Circular folds and villi are absent in the colon. Microvilli projections of membrane membrane 1 micrometer higher than that extend from the apical surface of the intestinal cells further increase the surface area for digestion and absorption by approximately 600 folds. Microvilli and the associated glycocalyx establish a brush border structure that present a considerable catabolic barrier of enzymatic activities. Mucus coating the epithelial surface of the gastrointestinal tract contains mucin glycoproteins, enzymes and electrolytes. These mucus is secreted by the goblet cells that are intercalated between enterocytes covering the villi as well as from the subepithelial glands, and acts as a potential site for drug interaction and binding. The pH of various intestinal segments range from slightly acidic to slightly basic. A slightly acidic microclimate at the immediate surface of enterocytes has also been described in the upcoming presentation you will be seeing here. In some cases, these variation in the pH might impact a drug stability or solubility or both. Now this transport, when we talk about the transport across the epithelial barrier, as we have just now have elaborated some of the physiological characterization of the intestinal barrier epithelium. The various transport processes that are used by the delivery system to cross the epithelial barrier lining are enlisted below, like the carrier mediated process, the passive diffusion process and the endocytosis mechanism. The polar molecules can easily diffuse across the tight junctions of the epithelial cells, which is known as the paracellular route. Molecules of less than 10 kilodaltons are absorbed from the nasal epithelium into the systemic circulation in sufficient amount without the need of any extra added excipients like permeation enhancers, while larger molecules like proteins, human growth hormones, etc. require permeation enhancers, basically like chelators, EDTA, citrates, salicylates, etc., as well as certain bioadhesive substances to increase the retention or the bioavailability of the delivery devices at the particular location. The factors which affect the absorption of the drug from the GIT are like pH, enzymes, microflora, transit time and the surface area. The next parameter of the biological uptake process is the extravasation. So, we will be discussing about the extravasation. Now, this extravasation basically it is a process which helps it's a process where you have both exposure of extravascular extracellular or extravascular intracellular process. So, for a drug to exert its therapeutic effect, it must move from the central circulation system and interact with its extravascular extracellular or the extravascular intracellular targets. This process of transvascular exchange, where the molecules or the delivery system, they go from the extravascular to the extracellular or from the extravascular to the intracellular target, that is within the cell organelles. This process is known as the extravasation. Now, this extravasation process is governed by the following parameters, that is permeability through the blood capillary walls, rate of blood and the lymph, as well as the molecular size of the drug, shape and charge of the drug. Now, when we talk about the blood capillary walls are basically three types of blood capillaries are there, that is the continuous blood capillary, fenestrated blood capillary, as well as the sinusoidal blood capillary. The continuous blood capillaries are basically common and widely distributed throughout the whole body, having tight interendothelial junctions and continuous uninterrupted basement membrane. In case of fenestrated blood capillaries, they have interendothelial gaps of 80 to 20 nanometer at regular intervals, whereas the sinusoidal blood capillaries, they have interendothelial gaps of 150 nanometer at regular intervals, along with the absence of basal membrane mainly present in the spleen, liver as well as bone marrow. In case of lymphatic uptake, whereas the after this is the fourth process, this is the fourth biological process, that is the lymphatic uptake. In case of lymphatic uptake, after extravasation, the drug molecules can either reabsorb into the bloodstream directly by the enlarged endothelial cell pores found mostly in the tissues, or enter into the lymphatic system and return with the lymph into the blood circulation. So, the various delivery systems which are administered as intramuscular, intraperitoneal, subcutaneous, transdermal routes reach the systemic circulation via these lymphs. But this lymphatic system is of minor importance sometimes, like when the lymph flow, because the lymph flow is very slow, as well as the lymphatic vessels are less accessible than that of the blood capillaries. But this route avoids basically the first pass metabolism. Compounds of higher molecular weight of more than 16,000 kilodaltons can be absorbed by the lymphatic transport. So, basically these 2 points can be considered as the advantage of this type of lymphatic uptake. The fifth biological process, that is the reticuloendothelial system. This is basically composed of mononuclear phagocytic cells which originates from the bone marrow. These phagocytic cells along with the bloodstream pass into various tissues and differentiate as macrophages. These macrophage can easily be targeted by opsonization technique. So, with this I come to an end to the presentation of the biological processes that take place or that are basically involved in the transport of the drug through the various ways. I hope you have understood this particular topic, and if you have any doubt, you can call me or you can mail me. Till then, take care and stay safe at home. Thank you.